Okay, genetics. Now we had a look at the big cow the other day. We mentioned this the other day, that genetics is the study of hereditary and how things are inherited. This is a very, very important idea. For most of human history, people, they could see that the children looked like their parents. They didn't know why that was the case. Okay? For most scientific history, they didn't know the molecular basis of how inheritance happened. And it wasn't until 1953 with Watson and Crick, they were able to demonstrate, look, it's DNA is that molecule that is responsible for inherited characteristics. And genetics has many, many, many applications in today's world. Of genetic disease, as you know from Year 9 science, disease can be caused by an external pathogen of some kind. It can be caused by a genetic condition, and we look at those, or it can be environmental. Poor nutrition or breathing lots of coal dust can cause. Those are the three causes of disease. For most of human history, the whole genetics thing, you know, it didn't exist in terms of how people considered disease. They just thought God, whatever God it was angry with you for some kind, and that's why, I don't know, you've got 3 eyes or maybe 1 eye or something like that. Okay, now those were the key learning intentions as per the Australian curriculum, okay, with key terms. And we're going to go through each of those. You'll see them as we go through the slideshow. I don't need to read that, but you do need to understand. Like, students always ask me, what's on the assessment? What am I going to be asked on? I tell you point blank, it's right there. In this slideshow, we cover those things, and on the exam and the test that I'm going to write for you, I'm going to go down that checklist. Did I write a question about that? Did I write a question about that? Yes, 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 yes. And when I've reached the end of that checklist, then I know my test is good. I also gave you the key learning intentions at the start of the year, okay, on that document, the paper document I handed out, and the electronic version is also available online. So you can look at those and go, oh, for genetics it's about that long. Okay, it's the big one. There's a lot of them, but those are what the questions will be about. So if you, long story short, if you make a cram card on each of those things that answers them thoroughly, you'll be fine. You'll have no problems at all. Okay, and those are the key terms that we're talking about. So each of these will be marked in red. You'll see them as we go through. And you need to highlight those as being examinable, as important. This is the language that we use in genetics. It's not kind of like this and sort of like that. And science students typically speak in generalizations. And what I'm saying to you is you can't. These are the words that will be used in the questions, and those are the words that you need to use in your answers. Okay? So we'll get better at that. We've got 7 weeks to get on top of this. Now, that's a simple picture, but it covers a lot of ground. Okay, so we'll drill down this a little bit. Now, you know, you've heard about DNA. Fine. You know DNA is the molecule of inheritance. Fine. Mrs. Green in Year 7 and in Year 9, you talk about things that are alive. You know, what distinguishes them from things that are not alive. And in Mrs. Green, they don't mention DNA, but they should, because all living things have DNA. Okay? And some people go, but if they're really clever, they'll say, but what about viruses with RNA viruses? Well, viruses aren't alive. So all living things have DNA. And what do we mean by that? Now, let's start with the biggest and work our way down to the smallest. This is a cell. What's at the centre of the cell? What's the structure that we say is at the centre of the cell, if you like? It starts with N. Nucleus. Now we're getting somewhere. Great. Now what are these strange little X-y looking things in here? Now, yeah, well, chromosomes, yeah, well done. What's happening here, when people think about DNA, maybe you've seen pictures of that double helixy thing, yeah? And they often think that the DNA is just this big, long, and you'll sometimes hear people say, oh, the DNA in each of your cells is 2 meters long, which is massive, and it's all coiled up. It's packaged in a way that's incredibly exotic. You have no idea. And I'll show you a video of that. But the DNA isn't one long thing packaged. It's actually broken up into chromosomes. Okay? And these are what the chromosomes look like. Now, that's actually these are what the chromosomes look like in terms of, well, what we're talking about that now. But you'll see these little wiggly lines here, and that's the DNA actually packaged up. And if you open up these chromosomes, you'll get this spirally structure. Now, multiple choice section, question 1. How many chromosomes do humans have? 46. Now, I weep for the future because so many times students misremember that. Okay, you have 46 chromosomes. Now, those chromosomes, if you want to impress me on that slide, put 23 pairs of chromosomes. They're matched pairs. So, guess what? No surprises here. You got 23 from your biological mother, 23 from your biological father, for a total of 46. Okay? That is the basis of sexual reproduction. Now, 2 metres of DNA, 3 billion DNA subunits, basis, ATGC. We're going to drill down into this a little bit. These are the nucleotides. But if you want to think of your DNA being like a cookbook, okay, the recipe for you, it's actually 1,000 telephone books thick, okay? It's a big cookbook. And that's the recipe for you, okay? And what you have is it's broken up into maybe 25,000 chapters, okay? Or 25,000, yeah, we'll say 25,000 words would be a better analogy. And each of those words, instead of having an alphabet of 26 letters like we use in English, we only have 4 letters in the genetic alphabet. A, T, G, and C. So the words can be quite long. And what are these words that we're talking about in this grand recipe? Well, we call them, really they're genes. Okay, so what you start with is the nucleus contains your DNA, but that's actually broken into 23 chromosome pairs, and the chromosomes themselves have a total of about 25 or 30,000 genes. Now this is really important. What do genes do? What is their function in the life that we see all around us? Genes express proteins, right there. And proteins have functions. Proteins, like your hair, could have a particular structure that we recognise as being hair-like. Okay? You are a big bag of proteins. Your DNA expresses proteins. You've got about 100,000 different kinds of proteins. They can be quite complicated because they can combine, different genes can produce proteins that combine together to make quite large structures. Okay? So, long story short, nucleus contains 46 chromosomes, which consist of 30,000 genes, each of which leads to the expression of a protein. That's genetics in a nutshell. Now, one little extension question. Oh, bit of UV comes down. UV radiation comes down. Dun, dun, dun. We've got our little C's, A's, G's and C's, our nucleotides. I've just disabled that section through UV radiation, high energy radiation. What is going to be the outcome of that change? I've changed a letter or 2 letters in my word. What's going to be the result of that? Downstream, yes?
Will it change the protein?
Bingo. And now you've got a disease. Okay? Now, maybe this changes the sequence, it changes this sequence and the protein changes a little bit, but it still works. So we're okay. Maybe it changes the protein a lot, but that gene is not really important in the grand scheme of things, and so you've got another copy from mum, so you're okay. It's still getting plenty of good protein through your system. Maybe that gene is responsible for preventing cancer cells from forming. That's a really, really bad roll of the dice. Okay, and now you've got a skin cancer. So remember from year 9 science, in physics, we looked at the spectrum, and we looked at the different wavelengths of light, and we looked at UV and X-rays and gamma rays, and we said they're high energy, and they're dangerous because they do that. They cause damage to DNA, which results in problems downstream. Does anyone have any questions about that one? Very simple slide, but a lot of ground covered there. Makes a good crown card. Okay, if you can get your head around that. Now, we looked at this just the other day. DNA is deoxyribonucleic acid, as its proper name. It makes copies of itself. So that's one of its jobs. That's how you start with sperm meets egg, and you've got a single fertilised cell, and then it makes a copy of itself, and now you have 2. And the DNA has made a perfect copy. And now you've got 4, and then 8, and then 16. It takes a long time for that process to finish off, to reach maturity. Obviously, when you're born, you're only this big and you have a few cells. And by the time you get to 18, when you stop growing, or 16, when you stop physically growing, okay, then you've got a lot more cells, about 37 trillion cells. A complicated process. Okay, so 1, it self-replicates. 2, it carries genetic information. Highlight those are its 2 jobs. Self-replicate, carry genetic information. Now, is the mechanism by which inheritable characteristics get from one generation to the next. Okay? It's not proteins, it's not voodoo magic of any kind, it's DNA. That's the molecule that has that job. The word genome means all of the DNA found in the cell of individuals. So that wasn't on the previous slide, you can put it there if you like. So all 46 chromosomes, your 46 chromosomes in total are your genome. Okay? Okay. Now they are packaged into chromosomes, which in turn are made up of genes. And we'll look at each of those specific things. Now, why do children look like their parents? Well, your physical appearance is determined by what? Your DNA. And where did you get your DNA from? Your mother and your father. That's why children look like their parents. But what is interesting is, father-son, complete strangers. Don't even live in the same country. This photographer travelled the world looking for people who had living physical clones, who just by chance have, in terms of the genes responsible for physical appearance, have a similar gene set to other people in the world. This girl met the other girl in a nightclub by chance, and they just happened to be at the same time about the same age, and boy do they look similar. Okay? And that's why you can have, it's not surprising that you look like parents, children look like parents, but it's also possible for people who are not related or closely related to look pretty similar to each other. Now, this is important. Chromosomes. Thread-like structure of nucleic acids, obviously the DNA. Remember, DNA is deoxyribonucleic acid, okay? And protein found in the nucleus of most living cells. Not all cells, just most cells. Red blood cells, for example, don't have chromosomes in them, okay? Carrying genetic information in the form of genes. They contain all the heritable information needed to construct an organism. Now, you know this already, humans have 46 chromosomes, and here's a new word, homologous, which means same size, same function. Okay? That's a new word. We have 23 homologous pairs, one from each parent at the same, and so homologous, that's what little asterisk is, same location, same function. Now, you've lucked out. In terms of, here we have our chromosomes here. This is a karyotype, which is a photograph. You don't need to know that necessarily, but it's a photograph of chromosomes that have been stained. And you can see the patterns are identical in terms of where banding is occurring and whatnot, right? Now, the biggest chromosome was given the name chromosome 1. Woo! That seems pretty logical. It's not A742, which is like horrifying for students. It's just 1. It's the biggest one. One from mum, one from dad, biological mum and dad. Chromosome 2, slightly smaller, one from mum, one from dad. Okay, so you can see here, all the chromosomes are not the same in their size. Okay, so this one has the most genes, and these ones down here have the fewest genes. Fascinating. So, you have, how many chromosomes do humans have? 46. Is there a problem with this karyotype?
Yeah.
Yeah, what's the problem? He's got more. Define more. Never use the word more or less if you've got a number.
He has
Sorry? He should have 46. How many does he have? 47. Well done. There's a triplicate down here. Now we've got trisomy chromosome 21, Down syndrome. Yes?
I was just going to say this how many has Down syndrome.
Yeah. 21 is Down syndrome. Now, how do I know that this individual is a boy? XY. Well done, if you've clicked onto that. There are the sex chromosomes. XY is male. And what's the code for female? YY. So write that down. YY is female. There's a lot of information here. Okay? So, there are our sex chromosomes. Now, an allosome is the name for sex chromosome. An autosome is a non-sex chromosome, and a somatic cell is a non-sex cell. Those are just terms. Okay? So you have your sex cells, and every other cell, skin, everything else, is called a somatic cell. You have your allosomes, which are the sex chromosomes, and autosomes for non-sex chromosomes. So if you want to put a box around this and put autosomes, fine. And then just down here, here are your allosomes, sex chromosome. What do you notice about the X and Y chromosomes? One's bigger than the other. That's really important. 1 in 15 boys are colourblind because the gene that they inherit from their mother is dysfunctional in terms of cone production in the eyes, and so it doesn't work, and they don't have a copy from their father. There's a few genes that males carry that only come from the mother, and they don't get a copy from the father because of this difference in size. And now you're starting to understand genetic disease, why sometimes a disease can be, the mother is classed as a carrier because she carries the genetic condition, but she can see colours perfectly, but has a son who cannot see colours because of that discrepancy in size. And we're going to go through that as well, but I'm just touching on everything. Any questions about that slide there? Yeah?
Is there a way that you can tell if your son or daughter will be colourblind?
You would have to do carrier mapping, which is when you have a fertilised, your sperm meets egg, now you've got your fertilised egg, and you let it divide up to a few cells, then you take one of them, extract the DNA, multiply it up and do genetic tests. But that's invasive. You're sticking a needle into, effectively, an embryo at a very early stage to extract a cell, and for colour blindness, probably not going to do that.
Yeah.
But for other genetic conditions, and maybe there's a... And do you really need to go through all of that? Or what's another way of achieving that but not endangering the embryo? What's a way of finding out if you need to test the embryo? Test the parents. Yeah? So you can just test the mother and the father, and this is the world that you're moving into now, where eventually, effectively on your Medicare card, you'll have your whole genome. Yeah? And there's a community in the United States already where they have genetic counselling because they have a high incidence of Tay-Sachs disease in that community. Boy meets girl, they fall in love, they've been going out for about 3 months. I think this is a pretty good relationship. They go to a genetic counsellor and they will get their genomes will be checked for 10 very nasty genetic conditions. And the counsellor will come out and tell them, I'm sorry, you have a 50 or a 75% chance of your children having maybe Tay-Sachs, which is brutal. And that community has not had a single child born with Tay-Sachs since they started that program. They don't believe in abortion, but they have embraced genetic screening and counselling. So the couple, if children are really important to them, either break up, or they adopt, or they don't have children. They're not forced to make that choice, but no couple has elected to roll the dice on that. Yes?
Would they be allowed to have a child?
Yeah, they can. Yeah, absolutely. They're in. Well, Tay-Sachs is a very, very, very bad genetic condition, and parents live in fear of those sorts of things. So that's just the nature. So that's a world that you're going to go into, for in terms of screening, will be much more common. Okay. Now, Down syndrome. There we go. Boom, boom. An extra copy of chromosome 21 from the previous slide. Down syndrome is the most common genetic condition question that I have seen. Okay, they 9 times out of 10, if you're going to throw a genetic question in terms of an additional chromosome or something like that, Down syndrome pops up. Yes.
What happens if you have a chromosome missing?
Most human fetuses that are conceived are not born. So 50 to 80% of the time, when a woman becomes pregnant, after a short period of time, a chromosome is missing or something like that, there's an error in this replication process, the fetus will self-abort. So, and this is early on, not 3 months or 6 months into the pregnancy, early in the first few days. And so the pregnancy is not even detectable at that time. Okay, so there is error checking going on. So I've known people who have conditions where a part of their chromosome, of a particular chromosome, is missing, and they're okay, they're fine. But I'd be rolling the dice if I said chromosome 1 missing, you'd be fine, but chromosome 2 missing, it would not lead to a viable pregnancy. There's a lot of error checking going on. Yeah, so parts of chromosomes can be missing. Occasionally, you get additional chromosome copies sneak through. So the Kenyan forest boar is a species of pig that has 2 extra chromosomes compared to every other species of pig, and it's doing just fine. So stuff happens. Genetic condition, disease or condition caused by a chromosomal aberration, which is, an aberration means something different to what is normal for the species. Extra copies or too few copies. You can have an absent or defective gene. So a gene could be missing entirely. Parts of it could be missing. There could be extra parts added to it. You see where I'm going here? Just a change in genetic information. This is called a hereditary disease because if it can be passed on to the next generation. Yeah? And that's how genetic diseases pass through. There's about 6,000 genetic disorders. Many of them are severe or fatal, but most often fatal later in life. Why? Why is that the case? Yep.
Because it's when they start to develop.
But why would it be more likely that a genetic disease would be fatal later in life than as an inheritable condition? Hint, hint, inheritable. Think of this. If a genetic disease was really, it was a dominant condition that killed everyone affected with it before the age of 2, how many of the next generation would inherit that condition? Well, no one, because no one can survive to get old enough to reproduce. You follow where I'm coming from? So if I had a genetic condition that resulted in my heart stopping when I turned 50, I've already reached an age where I'm most likely to have reproduced. That's a pretty bad roll of the dice, but you can see how that could be passed on to the next generation. So that's typically why you see them being fatal much later in life. This British boy, we've talked about him, he's immune to 200 genetic diseases because his mother had him karyotyped. And that was only a couple of $1,000s to do. That's a new technique that will be generally available to everyone within the next couple of years. And so this is the sort of world you're going into where we're thinking of starting a family. Is it going to be like Gattaca? Are you going to go see your local geneticist? You know, are you going to pick a candidate child? Okay, now, this is a cram card slide. On some of my slideshows, I put the letter C up here for cram card because you just have to know it. Okay, this one is solid gold. Slide 9. Okay, a gamete is a sex cell. It can be a sperm or an egg in animals, pollen or ovules in plants, but typically we're quite gentle to you, and we have, when we talk about sex cells, we talk about sperm and egg. Now, a spermatozoon or sperm is the mature male sex cell, usually has the head and the flagella. Okay, it fertilizes the ovum, which is the female sex cell. Okay, now let's zoom in on the head of this sperm cell, and what do we see? If you had to count, if you had to guess, how many little liney things do you think would be there?
23.
23. Well done. Well played, sir. 23. This is my very primitive drawing of the chromosomes. So the sperm carries 23. How many does the egg carry? The ovum. 23. Sperm meets eggs for a total of 46. Get that right. How many chromosomes do my skin cells carry? 46. How many chromosomes do my eyeball cells carry? 40. My liver cells? 46. My kidney cells? 46, 46, 46, 46, 46, 46 for all of my cells except sex cells. 23. Yes?
If you had an extra one, which would come from your mother or your father?
Extra
Chromosome.
I couldn't tell you. And there's a fascinating article in the paper today, well, a paper from overseas, where they talk about a shrimp had carried a mutation and it has 3 sets of chromosomes. So one shrimp had a very weird sex cell where it had a full set of chromosomes, you know, in there, and it reproduced offspring with a normal shrimp and produced a viable offspring that had 3 sets of chromosomes, didn't die, that can't have sex, but can lay eggs that produce cloned shrimp. So all of the eggs produce female babies, all of which carry the mutation and which can grow up and then produce eggs. So it's asexual reproduction. Okay, and they're starting to dominate in Europe. They're very tough little shrimp. They have a weird genetic mutation. The species is 25 years old. That's it. It's a new species. It just popped into existence. Okay, now, haploid, highlight those 2 letters. Haploid for half. Got it? Haploid is half the number of chromosomes. How many chromosomes do humans have? 46. The haploid number is 23 chromosomes. Okay? Diploid, okay, complete set of paired chromosomes, 23 pairs. Human sex cells are haploid gametes, each of which contains 23 chromosomes, and here is where rubber meets road. A haploid gamete, what does haploid mean? Half. Gamete means sex cell, sperm cell, containing 23 chromosomes. 22 autosomes. What are autosomes? Non-sex chromosomes and 1 allosome, a sex chromosome. 22 autosomes, 1 allosome. Here's an allosome, a sex chromosome, can be X or Y. This sperm cell carries X, this sperm cell carries Y. And whereas eggs only have X. Okay? Egg cells only carry X. So the sperm cell determines the sex of the baby. Whichever one gets there. So if this sperm cell fertilizes the egg, little boy. If this sperm cell, little girl, XX, X from the sperm, here's the egg, carries X. XX in the sex cells will be a female baby. XY from these 2 sex cells will produce a male baby. Okay, so that's it. Any questions there? So, a lot of ground covered, but important. Yes?
You said XX equals
Female?
so does that mean YY equals a female as well?
No.
You can't have Y.
What? Nah, why can't you have YY?
Because you always start with eggs only have X.
Yeah, that's really important. That's a great trick question. Okay? Like the egg cell, this is not to scale, egg cells can only carry X, so the sperm cells are X or Y, so the only possible combination is XY and XX, you say, yes? How simple that is. Oh wait, sometimes you get XYY, sometimes you get XXY, weird things happen.
How can you get 3?
Okay, you know, who knows Dwayne Johnson?
Yes.
And why do you think he's a big boy?
X, Y, Y.
What do you think?
Danny X
What do you think that the Y sex chromosome only codes about 50 genes? What's an important one in terms of masculinity that is coded on the Y?
Well, this isn't the answer, but like, would you say that XYY or YXY, that's not a genetic mutation, is it?
It's a mutation of sorts, yeah. What do you think, of those 50 possible genes, what's a male-ish protein that the Y probably produces that's related to maleness?
Hormone.
Testosterone. Now what the heck happens if I have 2 functioning copies and I'm jacking out twice the amount of testosterone that I would normally? You're going to muscle up. You're also probably going to be really hairy. Okay? And that's male XYY. That's female. This happened a few years ago. Female Olympic athlete who was more muscly than you would expect, and because she was producing more testosterone than a female normally would, so she muscled up more readily. So she's genetically a female, and she will go on to produce viable children, but she has 47 chromosomes. So stuff happens. Yeah? So there you go. Genetics. But, yeah, Danny, that's a very, very important point, that because the egg can only contribute X's, then the possible offspring can only be XY or XX, and never YY. Now, some textbooks show chromosomes as double strands, and that's what the picture on slide 3 looked like. Okay? What are these chromosomes doing? I mean, that's what a chromosome looks like, but this chromosome here is making a copy of itself. Okay? This chromosome here is making a copy of itself. So, look at this simple cell. How many chromosomes do we have in this simple cell?
6.
No.
7, 8.
I have seen 4. Let's go to the start. Okay? It starts with effectively 1, 2, 3, 4. Right? So you've got 2 long ones and 2 short ones. But then they make copies of themselves, so you get this look of the thing, and then that gets pulled over to that side, that gets pulled over to that side. And what we're starting to see here is cell replication. Okay? And we discuss this later. So you start off with the chromosome. So how do you, when a cell is making a copy of itself, it's going through this process here of mitosis, where each chromosome makes a copy of itself, they get pulled to the sides, each copy gets pulled to the sides, then you get a division, and now I've got 2 cells. And it goes from there. So you need to recognise that if you see this sort of structure connected in the middle, that's a single chromosome. Because some textbooks will show this. So how many chromosomes do I have? 6. 1, 2, 3, 4, 5, 6. Okay? That's just a different way of representing the information. But sometimes you can be asked, what is the cell doing at this stage? Right, now we define what the gene is. Okay? The smallest heritable unit of DNA. Now, very importantly, it's a unit. Because as a unit, it has a function. If you break it, it's no longer functional. Okay? So, it's the smallest inheritable unit of DNA, consists of a sequence of base pairs. We'll talk about that later. But the base pairs are G, A, T, C. It carries genetic information for a single character, encodes a protein or an RNA product. Okay? That's what genes do. For you guys at this level, we typically say proteins. So you start with DNA here, and you finish up with a protein, and the protein has a job to do. Okay? And it might be a job like carrying oxygen in your blood, or it might be a job mopping up damaging chemicals that you've somehow picked up in your diet, or just keeping your circulatory system clean. There's 1,000,001 jobs that your body's doing, and the proteins are doing that. It might be an enzyme. So many proteins are enzymes because they're biological catalysts. They let things happen fast. Okay? Without them, you couldn't function. Okay? Now, single gene traits include tongue rolling. Can you roll your tongue? If you can, you've got one copy of a gene that allows that to happen. 1 or 2 copies, but only one is needed. Okay? So this is easy. These are single gene traits. Widow's peak, that's when the hair comes to a point, like the old Draculas. Yeah? Wet earwax. Okay, we're going to do an earwax sample. No, we're not doing any of that. Okay? Wet, sloshy earwax is a single genetic trait. Photic sneeze reflex. I thought everyone could do this. I've got this one. Only 35% of the population does. That's my X-Men power. If I feel a bit sneezy, I look at a bright light and I sneeze.
Yeah. I've got it.
Oh, yes, I got it.
It's not... Put your hands up if you've got the best single gene. See, there you go. Bang, 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 bang. Photic sneeze reflex. Only 35% are gifted. Fit. Guaranteed to bring in the ladies. So, lucky. Attached earlobes. Has attached earlobes versus not attached earlobes. Okay. Chin fissure, like that. Yeah, you got that little dimple. S-methylthiester detection is recessive. Okay? So you actually need to have, you need 2 copies of it to have that. And your mutant X-Men power is you can detect the smell of broccoli in human urine. That is not... there we go. Mutant.
Say it again.
You can detect.
You can detect the smell of broccoli in human urine.
I can detect the smell.
Well, you're thinking broccoli. How is this useful? But what I'm saying to you, no, not broccoli. Brussels. No, hang on. Asparagus. Not broccoli. Asparagus. Now, you're saying that's not a very useful mutation. Like asparagus is not a common vegetable. But what I'm saying to you is your nose is slightly more sensitive in a certain area of the spectrum. That's all. Which, nowadays, having a great sense of smell doesn't really help you in a great sense because we don't have a lot of predators. But, so our sense of smell has actually degraded dramatically compared to humans 100,000 years ago who had highly functional smell because it could be important in terms of life or death. Is food fresh? Picking up different chemical scents, potentially escaping predators. Pigmented iris, any colour but blue. Okay, so eye colour. Freckles, polydactyly. Does anyone have more than 5 fingers or toes? No, you don't have to. Don't have to put your hand up for that.
I have 10 fingers and 10 toes.
My cousin has webbed feet.
That's another one. Let's see, polydactyly, dimples, woolly hair, long eyelashes greater than 1 centimetre, short big toe. So, now, here we go. Now, very importantly, here is a gene, here's a chromosome, and what you have, it consists of DNA, and what we have is different, this part here is a gene on that chromosome, and it codes for a protein. This part here codes for a different protein. This is what we call junk DNA. That doesn't code for a protein, but as it turns out, is probably really, really, really important. Okay? So, genes are those kind of things. There's another little bit on genes on the next slide. Okay? Highlight that point. Heritable unit of DNA codes for a single character, and we say the locus is the location on the chromosome of the gene. Determine the heritable features of an organism. So what is the smallest unit of inheritability? The gene. What's the largest unit of inheritability? Genome. All DNA. So genome is the largest. What unit of heritability do you have 46 copies of, or versions of? Chromosomes, yeah. Okay, so they're all made of the same deoxyribonucleic acid. Genome is the total, chromosomes are the 46 big units, big collections of genes. The gene is the smallest one. Now, here is a tricky word. Allele is different forms of a gene. Okay? So, for example, with eye colour, we just said eye colour is a single gene trait, yeah? But there might be 22 different versions of that gene. You follow? Like, okay, let's talk about cars as cars being like a gene that most houses have 2 cars. Don't get caught in minutiae, but we'll say, oh yeah, so that house over there has 2 cars. 1 car from mum, 1 car from dad. That house over there has 2 cars. 1 car from mum, 1 car from dad. But are there only 2 types of cars in Bendigo?
No.
No. Each house can only have 2 cars, but it could be Toyota RAV4 and a Passat of some kind. And that car over there has, oh God knows, a Hyundai something or other, Sonata, and, you know, something else. Every car can only have 2. Every house can only have 2 cars, but there are many different versions of cars. So for Gene TomTom, I'm just giving this random name. For Gene TomTom, how many versions of TomTom, how many copies of TomTom do you inherit? How many copies of the TomTom gene do you inherit? 2. 1 from your mother, 1 from your father. But I might have TomTom 1, TomTom 2 genes. Chloe might have 2 copies of TomTom 1, okay? But it might go, Angus has a copy TomTom 7, TomTom 6. And then it goes, every student in the class is different because they have different alleles. We all have 2 TomTom genes, but we have different versions of that gene. And that's what we're talking about here with alleles. And so you can see there are many eye colours because there are many alleles for eye colour. Now, a character is what we can see in the real world, hair colour and things like that. And a trait is the variant of a character. So we'd say the character is eye colour in humans. But these are all the different traits. Green, brown, topaz, kind of cool. Aqua, blue, grey, jade. So these are the exotics. Okay. So that is everyone really highlight allele, because that is a particular word that always comes up in assessments that students struggle with. Yes?
What happened with, like, you know, David Bowie's like 2 different...
Yeah, that's kind of cool, isn't it?
Yeah.
Yeah. That's, I believe, is a mutation that occurs after fertilisation. So I'd have to look it up. I suspect it's, you have this particular set of genes that are responsible for constructing organs and things like that, compound structures. And I suspect that it's a mutation in that gene, so the instructions it's giving out is different to that. I really have to look it up though. Yeah, but it is rare but cool.
The
Yes.
different coloured eyes also really common in dogs and cats, I think.
I don't know how common they are, but they are kind of cool to look at. Now, this slide here, whatever the number is a cram card. Okay, now, I'll tell you this point blank, you won't believe me, but I'm telling you about 20% of your genetics test is on this slide, and about 15% of your mid-year exam is on this slide. Okay? So it's important because we have this thing called a Punnett square. Okay? And you need to know how to draw these and how to interpret them. You'll get given pictures of them and ask questions, or you'll be given information and you need to draw them. That is going to happen. I cannot be any clearer. That is definitely going to happen because it's a key learning intention. You need to know Punnett squares. Okay? And because these questions tend to be quite big and they are hyper dangerous, because if you get the Punnett square wrong, then all the questions thereafter, you're going to be in error. Okay? And we're usually pretty gentle on you when we grade those follow-on questions. If it's true for what you've drawn, we'll give you consequential marks. But you really need to navigate these. And we will practice this. Genotype, genetic makeup of the organism, the genome, we talked about that already. But the genotype is based on the genes. The phenotype is the physical thing you see. I have genes responsible for my hair colour, but my hair colour is what you see. That's the phenotype, okay? The physical thing, okay? Now, genotype plus environment determine phenotype. For example, height is phenotypic, how tall you are. But if you grow up in an environment where there's very little protein, you'll be about ballpark 30 centimetres shorter than you would be with the same genes in an environment with plenty of protein. Okay? So I worked in South Korea, for example, in the 90s. Very common to see South Korean men about 5'1", 5'2", and their sons 6'1", 6'2". Okay? And that is based upon the environment. Phenotype doesn't usually indicate genotype. For example, we'll look at dominant and recessive genes, and so that's where this comes in. Okay? Now, when we talk about dominant genes, this is tricky for students, so get your head around this. We use capital letters. So these ones here we call dominant genes. The little p's are recessive genes, and when we're talking about a particular gene, the dominant gene, we choose a letter for that, any letter, but choose a letter. This is a bad example, bad me. Because big P and little p, if you're writing fast, kind of look the same. A much better choice would be big A and little a, because they're clear. So when you choose letters for dominant and recessive genes, choose letters like A or B. That's a good one as well. C is terrible. Okay. D is good. E is good. See where I'm going with this? R is good. Okay. Now, listen carefully. This is important. A dominant allele produces the same phenotype whether its paired allele is identical or different. Dominant alleles, boy, that word's important, mask recessive alleles. Now, what do I mean by dominant and recessive? Look, long story short, we're going to talk about flower colour, and the allele for purple flowers is dominant. The allele for white flowers is recessive. So purple, I'm going to choose the symbol big P. For white, I'm going to choose the symbol little p. What does big P mean? Purple, which is dominant. Little p means white, which is recessive. Now, here is, I'm going to say, here's my mother flower, here's my father flower. What is the genotype of the mother flower? Big P, little p. What colour is the mother flower? Not sure? It's purple because this copy masks this copy. So all we see is purple. Now, this is the genotype of the father. Big P, little p. What is the colour of the father? Purple. But they still have these there, but you can't see whiteness. Now, what possible offspring could this mother flower and the father flower produce? Well, we draw it up as a square, a Punnett square. Now, if the sex cell, if this is the sex cell and this is the sex cell that combine, we're going to get big P, a purple offspring. But wait, maybe we have this sex cell and this sex cell combine, we get big P, little p, still purple. Because big P, if you have even one copy of big P, you get a purple flower. Because it's dominant. Now, if you have this one and this one, big P, little p, what colour? Purple. But if you have little p combo, you get white. And this is why you can have a brown-eyed father and a brown-eyed mother, and they have a child with blue eyes. Follow? Because the mother and the father are both carrying the recessive allele for blue-eyedness. Because to have blue eyes, you need to have the double recessive. Yes?
Does that mean that if you have two parents with the same colour eye, there's a 75% chance that you can get their eye colour?
No, because it depends what they're carrying. If you can have, here, we'll do it for brown eyes. Look, here's brown eyes, two brown-eyed parents. And you need little b to have blue eyes. How many children will they have with blue eyes?
All of them.
Little b.
Zero.
Because there's no little b's going to the next generation. Okay? How many, here's another combo. How many children will have blue eyes from these two parents? You need little b. None. You can never get a little b combo.
It's just 3 big ones and
So,
1 little.
you can only have that situation, and at that point you have a 75% chance of brown-eyed children and 25% chance of blue eyes. This situation, 0% chance, this situation, 0. Yes?
If two people and they're from the same family,
Yep.
and they have a baby, what happens if they have twice?
Well, the problem is, if they're carrying a genetic condition, that it's likely that people who are closely related, there's a greater likelihood that they'll both have the genetic condition, and they could pass it to the child, and the child has 2 copies of a gene that's dysfunctional. And so that's why it's very dangerous. Yeah. So, listen carefully. Now, this part down here is straight from a test. Okay? Gametes, meaning sex cells, for the parents, you need to list these. Okay? Big P for parent 1, big P, little p for parent 2. Those are the sex cells that they carry. Now, the possible genotypes, what are all the possibilities? Big P, little p, little p. Now, what are the ratios? You read those off. You can have, this is the possible ratios, 1 like that, 2 like that, 1 like that. That's not a great example. Why? Because it's not clear. What you should have on your notes is 1 times big P, 1 times big P, 2 times big P, little p, and then 1 times little p. That is unambiguous. Just putting the numbers down is not clear. You can also use percentages, but you have to include the letters. Do you follow?
Yep.
You must include, write that down. You must include the letters. If you don't, you get 0 marks because you're not being clear. Okay? And then these are the genotypes, the genes of the offspring. These are the phenotypes, what you see in the offspring. Purple and white. Ratios. 3, again, 3 purple to 1 white. It's not enough to just put numbers. You must put what you are talking about. So either the genes or the colours. That is what you will be asked. Now, I've got 90 seconds. I'll cover these last things. A recessive allele produces its characteristics only when there are 2 copies. All of this means 2 copies needed if you want to see it. Yeah? All of this means even one copy will mean that that's what you see as the phenotype. Now, homozygous means identical. Big P, or big P, little p. They are identical. Heterozygous is non-identical. Now, I will show you later on, listen up for that last minute, I will show you later on a question, but the questions typically say something like this. A father who is homozygous for a condition marries a woman who is heterozygous for the same condition. What is the likelihood that they will produce offspring with the condition? And you go, huh? A father who is homozygous, well, he might be big P, or big P little